Once again, good day, good evening, from wherever you are joining us today. This is a full webinar into our program titled Introduction to Big Data Bioinformatics. My name is Aditya Boishade. I'll be one of the facilitators and mentors in this program. Today, we would like to give you inroads into the program. We'd like to talk about the program syllabus, program features, and how to get participants registered into this program. We also love to talk about biotech as a company and OmicsLogic as a program. But today, we are not the only ones. It's not only biotech. We are doing this in conjunction with biology solution, right? And in the next few minutes, we would like to invite the CEO of Biologics Solution, Dr. Voke Toye. As I said earlier, the program mainly is to introduce you and share with you details about this upcoming program and explain how the training resources we discussed today will be used in the upcoming Big Data program. I hope everyone can see my screen.
Yes.
Okay, thank you very much. So, Biotech was founded by expert mathematicians and people with expertise in the field of biology, in the field of mathematics, and in the field of computational science. Starting with Dr. Alfred Tauber from the Tauber Medical Institute in Israel. And since then, Biotech has been working with a whole lot of schools in more than 100 countries to teach them about bioinformatics from the basics. So they've been collaborating with a number of academic institutions around the world, and they have participated in content evaluation, curriculum review, project designs. So as a result of this, OmicsLogic programs have been thoroughly evaluated and are known to lead to excellent outcomes. Importantly, the programs are designed to support students and beginners with minimum bioinformatics background, including biology, biomedical studies majors, as well as faculties interested in bioinformatics, data science, and biomedical research. Part of these countries is Nigeria and Africa generally, where starting as early as 2019, Biotech with the OmicsLogic programs have involved Nigerian faculties. One of our partners who will be working with this program is Dr. Voke Toye, the CEO of Biologics Solutions. Dr. Voke, are you there? I would love her to introduce herself and talk a bit about our experience with Biotech. Dr. Toye.
Yes, good afternoon.
Okay, good afternoon, ma.
Can you hear me?
Yes, we can.
All right. Good afternoon, everyone. Thank you. I would like to welcome you all to this free webinar. My name is Dr. Toye. And as Dr. Aditya said, I'm the founder and CEO of Biologics Support Services here in Nigeria. Biologics has pioneered several trainings in molecular biology and bioinformatics with the aim of building required skill and capacity for the emerging biotechnology industry here in Nigeria. I have benefited immensely from the OmicsLogic training, and in 2000, Biologics has partnered with Biotech in offering the first big bioinformatics big data training here, virtual training for Nigeria. So it's well known that biology is now a data science, and we're no longer dealing with single cells. And as we generate more sequence data through NGS sequencing, there is therefore an increasing need to know how we would analyze this ever-increasing data in order for us to arrive at outcomes that will be of immense benefit in disease diagnosis, treatment selection, and in agriculture. So one of the key aspects of this training will be engaging our participants in basic translational research projects. For instance, you'll be able to learn how to use multi-omics approaches to interrogate genomic or transcriptomic data from cancer cells. So this has the potential of predicting the phenotypic changes of cancer cells and their responses in different therapeutic agents. So I'm sure that this training will be a new learning curve for many of us here, and it promises to be a rewarding experience for those of us that would go ahead and enroll for the 3 months training, 3 months training, I think. So thank you, everyone. Over to you, Dr. Aditayo.
Thank you very much, Ma. That's Dr. Vokitoye. She's also one of the facilitators and one of the mentors in this program. So we'll be hearing more from her. So in a nutshell, I'll basically just run through what the plan is for this program, what biotech is, and what to expect in the 6 weeks long program, right? As I said, this is part of our OmicsLogic program. And when we talk about OmicsLogic, what exactly is the goal of OmicsLogic? The OmicsLogic goal is to provide training in bioinformatics to enable independent research that are guided by mentors and peers, to develop a growing community of bioinformaticians with shared passion for data-driven research and appreciation of citizen science. Another goal is also to enable students, clinicians, and faculty of all backgrounds to develop novel and independent research using latest technology in life science and data science. What are our programs? Our plan is to have between 5 to 6 interactive sessions, starting from next week, Wednesday, November 24, where we'll talk about introduction to big data in biology. Then subsequently, the 2nd to 6th programs, we'll talk about genomics, introduction to genomics, introduction to transcriptomics, differential gene expression, introduction to biomedical, statistical analysis, and machine learning. And we plan to end with introduction to bioinformatics projects, right? We'll also introduce you to some projects before the end of this program. We have a plethora of local and international mentors, starting with the CEO and founder of Biotech, that's Elia Brodsky, followed by Dr. Mohit Mazonda, who has a PhD in computational biology. Joined by local mentors, myself, another pathologist and a consultant pathologist, and also in faculty. Then with Dr. Vokitoye, who is the CEO of Biologics Solution. These local and international mentors will guide us all through these 6 weeks. So again, what exactly are we talking about? What exactly is bioinformatics? Why are we calling it big data, right? Biology is rapidly acquiring the character of data science because billions of data points on genes, proteins, and other molecules are compiled in large files and are being systematically studied. So analyzing this data will lead to more knowledge, more understanding about living organisms. And we can use this knowledge to assist or understand human health, biotechnology, crops, and livestock. So this course is just to give an overview of this bioinformatics and the application of bioinformatics for students, as I said earlier, for beginners, and majorly for biologists and clinicians. With this influx of data with associated methodologies, people are now going into data-driven research, which makes bioinformatics for life sciences as prominent as the emergence of computers for the tech industry. In some years back, biologists, clinicians, doctors have to consult biostatisticians to be able to understand their data. But with the advent of technologies, now every clinician, every biologist, every life scientist, every medical lab scientist, every microbiologist is expected to be a data scientist. In fact, it can be said that biology itself is now a biology itself is now a data science, right? So there's so many resources out there, and the analysis of these resources and the data they give us tell us a clear story that as a scientist, as a researcher, as a student in life sciences, you must be capable of not only understanding the science, right, but you must also understand the data, and you must also have the opportunity to be able to do research with these data. So this program is designed to highlight the role of big omics data. You know, when we talk about big data, what are we talking about? You know, after the sequencing of the human genome, it is said that the human genome is about 3 billion base pairs. Imagine 3 billion base pairs. If you want to analyze this information, if you want to glean knowledge into this information, you cannot do this by mere mean, median, by t-test, by simple statistical analysis, right? This is big data. You need advanced statistics to be able to understand this. And this data, understanding this data is now shaping modern biomedical innovation, where the molecular data and the analysis is being used for precision diagnostics, big data integration, excuse me, and the machine learning are transforming personalization of treatment selection, coupled with innovative algorithms that now allow for unprecedented capabilities with drug discoveries and design of large molecules and drug repurposing. We all know that with the advent of COVID, before it takes 20 to 30 years before a drug is designed or vaccines are manufactured. But with the advent of artificial intelligence and machine learning, it didn't take us up to a year or two before we could get drugs that are working actively for COVID. That's one of the essence of bioinformatics, big data, and chemoinformatics. So at this point, I would like to pause and ask if there are any questions. You can put it in the chat, or you can unmute yourself and ask. Any questions? Please, can you all hear me?
Yes.
Yes, very.
Okay, so I should assume there are no questions for now.
Hello.
Hello, I can hear you, sir.
Hello.
Yes, sir. No question from our end.
No question for your hand. Okay.
Okay. From my end,
Yeah,
there's a question. Can you hear me?
I can hear you clearly, sir.
Okay. So how can this database be applicable in pharmaceutical society?
Thank you very much for that question.
Like in aspect of therapy.
Thank you very much for that question. In fact, I can safely say that more than any other aspect of healthcare, pharmaceutical industries are the ones that are most gaining from it, right? Imagine almost $5 billion of drugs are being spent, yearly on common drugs, on common antibiotics. But if you're a pharmacist or if you're a pharmaceutical line, you understand that pharmacodynamics and pharmacokinetics work differently on different set of people, right? The dose of amoxicillin that will work for me might not be the dose of amoxicillin that will work for you. And this is dependent on a lot of genetic factors. Imagine we are able to understand the genetic makeup of each person, of each individual. And because of that knowledge, you're able to tailor a drug only for that individual. How lovely would that be? That implies I don't need to waste money buying a plethora of drugs. I just need to buy one that I know has been tailored for me. That is what we call personalized medicine. That's one. Two, if you're a pharmacy or in pharmaceutical science, you also understand that before the invention of any drug, it takes years before you can finally get a drug that works succinctly for a condition. But now with the advent of bioinformatics, people are going into what we call chemoinformatics, where you can begin to design drugs. You just sit down on your laptop, you design drugs that work for each chemicals, right? And with that, you can easily get a certain drug for a disease condition in years, not decades. Not only that, it is also assisting what we are calling drug repurposing, which was very evident in the COVID pandemic. Imagine how many number of drugs that are supposed to be used for other diseases are now being used for COVID. That's drug repurposing, right? Bioinformatics is the key to that. Bioinformatics opens up to that. It opens up the genomics. You understand an individual's genomics, and you tailor a drug for that individual, personalized medicine. I hope that is clear, sir.
Thank you, sir.
So there's really no aspect of life science. When I mean life science, no aspect of biology, no aspect of molecular biology, no aspect of agriculture, no aspect of medicine that bioinformatics is presently not playing a cogent role in assisting treatment, molecular diagnosis, and precision medicine. So in a bit, we were talking about big data. I'm sure you've heard the word omics, right? And I'm sure by now you've heard genomics, transcriptomics. What exactly are these omics? And what exactly do they mean, right? This is one of the things we talk about in this program. We'll start with genomics, which is different from genetics. Usually when we talk about genetics, we're talking about the study of a gene or a pair of genes or some number of genes to study them and see how they work, probably their transmission to protein or in enzyme formation. When we talk about genomics, we're talking about the whole complete 3 billion pairs of the human body. We're talking about 3 billion base pairs, how to understand these 3 billion base pairs, how to glean information from it, and how to explain either a disease condition, susceptibility to an infection, or molecular pathogenesis of cancers, right? So this is a field that can assist in understanding why is Mr. A susceptible to cancer A, and why is Mr. B susceptible to cancer B. For a lot of years, scientists and clinicians actually thought breast cancer was just one cancer, was one cancerous. But with the advent of bioinformatics, they were now able to explain that when we say breast cancer, we're actually talking about 4 or 5 variants of different cancers that could affect the breast. This is one of the advantages of bioinformatics. Also, there's also a field known as transcriptomics, which will talk about gene expression. I'm sure you know that every cell in the human body consists of the same gene information, meaning the cells in your head, the cells in your leg, the cells in your abdomen, they consist entirely of the same set of information. Then the question is, why are cells in the head? Why are they expressing air? Why are the cells in the leg? Why are they expressing other things, right? This is what we call gene expression. And that's what we call transcriptomics. Understanding of this will let us gain information about, despite the fact that each cell contains the same information, how come some genes, some proteins are expressed somewhere while others are expressed elsewhere. We can also use this to understand tumors or tumoral conditions, right? You can take sample from a tissue and take sample from normal cell and then compare what genes are being expressed by this tumor condition that are not expressed in normal genes. And from this, you can actually glean information about the genes that are involved in that molecular pathogenesis or carcinogenesis. And you can then decide on getting drugs to either tailor drugs for this condition and all. So there are other things like the other omics, like epigenomics, like metagenomics. You know, metagenomics is the one that I feel it's very critical. For microbiologists, you will know that with culture, scientists still don't know about 75% or 80% of microorganisms we have both in our body and in the environment, because culture has a lot of limitations. But with metagenomics, we can gather or adequately analyze almost 90-95% of the microorganisms. And not only that, we can also see the function of the microorganisms, their effects on the human body, whether in the gut, whether in the brain, whether in the oral cavity, or their effects on the environment, right? And so that field is known as microbiome or metagenomics. So these are the multi-omics data we'll talk about. One of our sessions will be on metagenomics. Then not only do we know things, we also learn and talk about what are some of the challenges, when we're analyzing this omics data. So this online program, as I said earlier, will start next week, Wednesday, November 24, and we'll cover some of the important technologies and the applications in today's world. The participants will be able to access bioinformatics resources along with expert and mentor support to get help with learning process that will be available for 5 to 6 weeks of this program, right? And we'll also be able to learn how to glean information from biological data. So briefly, let me check if there are questions. Oh, population genomics. For most of us, we know that, especially when we're African, we work between 10 and 4. So all our programs start at 4:30 p.m., right? And usually last for 90 minutes. By 6, we will be done. Now, this program is actually an hybrid program. Our live sessions will be majorly focused on practical areas, right? Later when I'm talking, you understand that Biotech actually has a couple of educational resources, right? One of them is just educational resources, where you go to and learn important concepts, important terminology, right? Then the practical hands-on, we'll do the practical hands-on on during our live sessions. So it's not as if we'll do everything during live sessions. What we'll do majorly are practical hands-on. So we'll give you a week, we'll give you assignments, we'll give you courses to read during the week. Then we'll come here on Wednesday and talk about it. And for some that actually miss some sessions, all our sessions will be recorded. Like this is already recorded. So all our sessions will be recorded, and each session video will be sent to registered participants, right? Not only that, that's why I said we have local and international mentors. We have WhatsApp groups, we have Slack channels where you can always ask questions. I'm one of the mentors, and trust me, you can come here at any time of the day, right? To ask questions about the session which you don't understand. So that way, you won't miss anything per se. I hope I've been able to answer that question. So I said earlier, not only are we going to teach you about resources, but also open you up to projects, bioinformatics projects. On our website, we have curated data set for practice. We'll tell you some of the publications where we are getting this data set. For example, there's the middle precision treatment of breast cancer, which was featured in Genome Biology in 2013. The publication is very interesting because the authors assembled a data set of 50 different breast cancers, cell lines, that represent all the whole spectrum of breast cancer variations. They analyzed it, they did genetic expression, and they were able to also get drugs that were able to treat different types of breast cancers. Because as I said earlier, breast cancer is not just one cancer, it's different types of cancer. So drugs that were treating one were not treating others. So they were able to get drugs that were specific for each breast cancer. We'll also talk about that process. So right now, I would love to pause a bit to entertain more questions. If there are no questions, I would love to let us join me while I log in to our first educational resources, the Learnorglogiccom, and we'll be able to register and create our profile. Are there any questions before I do that? Any questions?
I have a question. Hello, can I
Yes,
ask a question?
Yes, ma'am.
I'm just getting a sense that this platform is all on the website. You guys don't train on tools within or outside of the platform?
No. When we get there, ma'am, I will introduce you to our main pipeline tool, right? The T-Bio server, where you have all bioinformatics algorithms. If you're familiar with bioinformatics, you know about Genomics, about FastQ, about FastP, all available bioinformatics tools. And not only that, also all machine learning tools, right? It's on that platform, we'll get there. But not only that, not only are we going to teach you this, we also know that for you to be an advanced bioinformatician, you need to be able to do some programs, right? And you know that 3 common programs for bioinformatics. You have OW, we have Python, and you have Linux. Also on our educational resources, we have OW and we have Python, right? And we'll also take you through the basics of this program during this course, right? You learn how, you're not going to learn how as a hardcore programmer, but you learn how that is relevant and specific of bioinformatics, right? So anything you need in bioinformatics, I can assure you, you get it within our educational resources and our artificial intelligence guided, smart, cloud-enabled bioserver.com. Is that okay, Ma?
Yes, thank you.
So we are not.
Okay, sorry, I have a question.
Yes, please, sir.
Okay, so thanks a lot for this program, and sincerely, I've been waiting for something like this here in Nigeria. I mean, it is the hub of molecular science currently. So I want to know how much is the full package from beginner to advanced, so one can plan a budget. You understand?
Yes, sir.
So how much is the full budget? Then two is will there be certification?
Yes, sir. Thank you very much.
And then,
these are things I'm planning to talk about, right? For this program, the way we planned it, we said is OMX Logic program. It's a step-by-step guided mentorship program, right? We understand that there are people that will be beginners, there are some that will be advanced learners, there are some that will be veterans. So we plan to meet everybody along the way. So we are going to hold a series of programs. This is just the starting point. This is more like for beginners, right? This is like just an overview for everything, right? And this program itself is $45 for this program. But our plan is starting from next year. We plan to hold a series of programs that we can move each person right from beginner to intermediate to advanced programs, right? We'll be holding programs in batches, in series, right? So that anybody, whatever level you are, you can join us through the program, right? Probably once in 3 months or once in 6 months or quarterly as the members desire. That's our plan. Do you understand, sir? Hello, sir?
Yes, I get you. But I want to also ask if hello?
I can hear you, sir.
Hello, can you hear me?
I can hear you clearly.
I got your point. I want to also ask if things like maybe DNA sequencing, DNA extraction, PCR, and all of those gel electrophoresis practices are also involved, because I know the organizers of this program they are kind of a biological practical based company. So I don't know if they do that, because many of us were just taught this thing theoretically. We don't know how to use this PCR machine, how to sequence a DNA, extract, sequence, and all of that. We're not given the practical insights into it. And I mean, we're just giving the theories, BCH this, BCH that, and we're not even taught how to do it. I don't know if there's a program like that that could come up.
Yes, thank you very much, sir. As I said, we as a company, we're actually into bioinformatics majorly. The dry lab, if you're familiar, everything you do in the laboratory, we call it wet lab, right? Those PCR, the sequencing itself, the electrophoresis, flow cytometry, they're done in laboratory, right? And that's why we partner with CEO Biologics Solution. If they also hold series of training programs on that, right? What we are doing as a company is, after PCR, when you get your sequence, what do you do with it? Right? So we are the dry lab aspect of bioinformatics. Do you understand? Biologics Solution, they hold a lot of programs, and we'll also talk about it. They will probably tell you when and how when they do all their own practical sessions. But for us, we are the dry lab aspect of things. Our goal is what to do after you have sequenced your data, right? What do you do with it? How do you analyze this data? How do you glean information from it? For some, PCR is just the beginning, right? When you do PCR, you get your data. What do you do with that data that you get after you must have done PCR? How do you glean information from that data, right? So we are the dry lab aspect of things. But biology support solutions they're actually chief. Their foremost job is actually on the wet lab of things, and they also hold practical training programs as regards that. Do you understand? But how we are, okay, Dr. Toy is raising, I think she wants to chip in and say a word or two about that. Dr. Toye?
Yes, I quite understand where the gentleman is coming from. So again, we see these kind of scenarios where many people still want to have a more basic understanding of where all this information is even coming from and how do we even get there. So like Dr. Adeta just mentioned, there is what we call the wet laboratory aspect, which is more hands-on on how you can actually get to your PCR and then your gene of interest that you want to work on and then get it sequenced, okay? But what they are talking about here is more of a lot of data that is already available in a lot of databases based on a lot of research that's taking place, especially looking at cancer cells, both with those that are there in the disease state or the normal cells. And so a lot of deep sequencing is done to get you data of a particular sample. Not only will it be genomic data, but also data that shows you the transcriptomic or the RNA sequence for that particular condition, as well as if you want to, the proteomic information in the data form, okay? And so that's why it's called big data. And so all of this can be analyzed using the tools available on the platform by Biotech. And part of this training will teach you or show you how these tools are used and under the different, using the different tools either for metagenomics or for RNA sequencing. You'll be able to take you through on how to use that. But for basic molecular biology skills that perhaps many of you did not even experience at the, during your programs, BSc or even MSc. Yes, Biologists definitely organizes trainings, hands-on wet lab trainings to address these gaps. And this is one of the things we do very, very well. I hope that helps you.
Thank you very much, Dr. Toye. I hope you understand that, sir. Hello?
Hello, please. I need to ask a question.
Yes, Mom.
Hello, thanks for organizing this program. It's actually been highly informative. Please, Dr. Toye, please can you share a link to those hands-on training, how I can access them?
Okay, so just let us have your email. Just Alfred will get in touch with you, give you additional information on that.
All right.
Alfred is also he's one of the participants here. He works with us. Okay.
All right. I will type my email in the chat box.
Yes.
All right. Thank you very much.
Thank you very much, Dr. Toye. And one of the things we're also planning is that it probably comes to a point where we'll have programs that will start with the wet lab details that Dr. Toye just mentioned, and we'll end with the dry lab details that we are talking about in bioinformatics, so that everything will be encompassing, right? These are things we are working on, and probably early next year or middle next year, we can make this happen, depending on what exactly the participants would love to have. So in a nutshell, let me just take us straight to our platform. I said we have a couple of educational resources. So I would love to start with the very first one. Just give me a minute. Can we all still see my screen?
Yes, we can.
Okay, so I will type the name into the chat forum now. The website is learn.omicslogic.com. It's already in the chat forum. So this is our educational resources. Here, everything you need to know about multi-omics, you can find on this website, right? It has So the first thing to do is to open a profile or register with this platform. I hope you can all see I'm typing it again into the chat forum, learn.omicslogic.com. And if you are new, if you haven't registered before, the first thing to do is to register. Either you click register for free. How many of us are on this website already? So if you're a new user, when you come here, just click on create an account, right? If you create an account, type in your name, your email address, and a password you would love to use. Not only that, you can also log in via your Gmail account or Facebook, right? So how many of us are here? If you're here, please can type one into the chat box. Are you already on this website? Thank you, Emanuela. The gift to your user already. Nice to know. You have some unfinished business with us. Yes. I hope during the course of this program, we'll be able to finish some of those users together. Okay, Samuel, thanks a lot. Mark, your user already good to know. So for the ones that have already registered, you can just sign in straight, right? For new users, you can just sign in now. Please, if you're having any issues with signing in, please just kindly let me know. So I'm already a user, but I'll just log into my account. Thank you, Samson. So even though this program itself is a paid program, I'm elated to let you know that there are a lot of freebies, a lot of free educational resources. In fact, our first 2 courses are entirely free. The only thing you just need to do is to open the website. I'm typing it again into the chat forum. Just click on that link, it will take you straight there. Learn.omixlogic.com, HTTPS, not HTTP, right? So when you come here, you can see this is my own platform. Thank you, Abubakar. You're currently on Bytes and Muku. Abubakar, you're the chief. Abubakar, you can attest to the fact that Introduction to Bioinformatics is free. Bytes and Muku is also free. So if you are here, the name you entered will show here. This is my own profile. And if I click on this, if you click on your name or your username, whatever you use, it will take you to your profile, right? And so that we'll be able to know ourselves, we'll be able to know who you are, we'll be able to relate with you. We usually ask that our profile is updated. If you select a file, you can put in your picture like I have, your name can be here. You can also give us your Facebook profile, because we also love you to follow us on our social media pages, and we also follow you back. You can put your link into these places. I can just give a small profile. For example, I was the STOA community manager for OMX Africa. I'm one of the mentors I said earlier. I'm a data scientist, a machine learning exponent, and I'm a pathologist. I'm also a faculty here in Bioinformatics Kano. I'm actually the deputy dean of the Faculty of Dentistry. So just a small profile was written there. So once you do that, you update this, right? And once you update this, everything will be obvious here. So we can learn to know you, we can learn to talk to you and network with you. And once that is done, you can just come here to courses. And where we have a plethora of courses. When I say plethora, I'm telling you we have a plethora of courses, right? And most of these, some of them are paid, but quite a number of them are also free. So the first 2 courses, especially for beginners, that will tell you everything about bioinformatics. The first one is Introduction. You can just type it here, Introduction to Bioinformatics. You see, it's coming as course 1. If you click on this, I can also send you this link. So that I just go straight to the course. I've typed the link in the chat forum. So when you come to this link, this course gives you the background of everything you need to know about bioinformatics, right? You can see it has 3 lessons. The first one is Introduction to Big Data Bioinformatics. It tells us about the application of bioinformatics in healthcare and translation bioinformatics, right? This is like the most basics. In OmicsLogic, we believe in taking you step by step We assume you're a beginner. We take you like a student. We guide your hands through the steps of bioinformatics. So that starts with introduction to bioinformatics. When you are done with this, and somebody asked about certificate earlier, we have different types of certificates. For each course that you finish, you get a certificate. Once you are done with these 3 lessons and you complete it, there will be questions, assignments after. Once you are done with it, you get a certificate for this course. So for each course you finish on our
I'm sending the number.
For each course you finish on our platform, you get a certificate. For every program do with us, for example, these are present introduction to biotech.
Let me type the account number to the school. Give your phone.
Can you please mute yourself, sir? Thank you very much. For this program, for example, big data informatics, when you are done with this program, you also get a certificate. So there are certificates for finishing each course, and a certificate for participation in each program. And for programs, the certificates are of 2 types, right? There is a certificate we call certificate of participation. For coming to this program, it's an exhaustive, comprehensive, hands-on program. And for each course you do, for each achievement you get points for it. So once your points are up to 3,000, you get a certificate of participation. If you don't make up to 3,000 points, we are not going to give you a certificate. But as I said, it's a practical hands-on program. But for some people that are very good, they are very motivated, once you get up to 10,000 points, you get a certificate of excellence. You become an expert. So during this program, if you're able to score 3,000 points, you get a certificate for it. If you're able to score 10,000 points, you are getting 2 certificates. You are getting a certificate of participation, you also get a certificate of excellence. This is coupled with certificate for each course that you're able to finish, right? I said earlier, the first course is Introduction to Bioinformatics. The second one, which also opens you up to the field of bioinformatics, is known as bytes and molecules, right? BYTS. You just type it here, and that's course 2. I also put this in the chat form so you see. So you can just click this link and it takes you directly to the second course. These 2 courses, as I said earlier, are absolutely free. They are absolutely free. You can just do them to just get an understanding of the way our educational resources are factored or modeled. So, any questions so far? As I said earlier, the 2 courses, bytes and molecule, and this, they are free, entirely free, right? You don't need to be a part of this program before you can do this. But once you're a part of this program, you have access to all the courses on these educational resources. As I said earlier, we have up to 15 courses of all multi-omics. We have projects, right? Projects that have been curated, that have been done. You can see these are projects, project topics for people that have written papers who break down the tools they use, right? These are projects. So we also teach you that. So you have access to all our courses, all our programs. Like I said earlier, for each activity on this program, you also get points for it. So any questions so far? Any questions? Please, can you still hear me?
Yes, we can.
Okay. So
Yes, we can.
Thank you very much. So if there are no questions, let me just go back straight to my presentation. So as I said, I've shown you the educational resources platform where you go and get the learning, right? You understand the theory,
I have a question.
Okay, sir. Go ahead, sir.
Okay, I want to ask, is there any specification for the kind of laptops we need? Because over time I do hear, okay, you have to get 8 gig RAM and you have to get maybe 1 terabyte because the installations you do might be heavy or something. Are there any specifications or?
That's a very nice question, sir. And that's where, when I talk about our next platform, that's where you actually understand why we are pushing this biotech. Now, when I started learning bioinformatics, I had to start learning by learning each of these bioinformatics tools, right? Meaning you have to do it on your laptop. Bioinformatics is computationally intensive, right? As I said earlier, the human genome is 3 billion base pairs, right? If you want to download the human reference genome, the last time I downloaded it, it was 25 gigabytes, right? Even if, especially you know the way Nigerian network is, even if you have data, you know that there is no likelihood that it won't be terminated at any point in time, right? So it used to be that heavy. But the way Biotech has gone around it is that our next resource I'm talking about is actually on the cloud. It's cloud-based, meaning you don't need to store anything on your system. Of course, you need to have a, Dr. Toye is raising up her hand again. Dr. Toye, ma.
Okay, I'll allow you finish with this before I speak. Just finish.
No problem, ma. So individually, without our next resources I want to talk about, if you're just trying to do bioinformatics and computation on your system, you actually need a system with a large hard disk, if possible between 500 gigabyte and 1 terabyte. And you need to have cores that are very fast because you are dealing with a large number of data. Remember we said this is big data. And for everything you do, you have to go through the processes of typing them or mapping them to the human reference genome, especially when you're dealing with humans. So it is computationally intensive. But the way around that is our next resource I'm going to talk about, the bioserver, T-bioserver, is cloud-based, right? So you don't need to actually do anything in your system. Everything you're doing is on the cloud. You just tell the system what you want it to do, right? Then dump it on it. Do you understand? You're not going to do it on your phone. You're not going to do it on your system. You don't need any resources whatsoever. Any laptop can do this because the only thing you're doing is once you've created a pipeline, once you've told the server what to do, you just log it there and just wait for it to do its thing, right? So because of our T-bioserver, you don't actually need any specification of laptop. Just a normal running laptop, a normal running network, and you're fine. Do you understand that, sir? Hello, sir. Doctor, you can go ahead, ma'am. It seems
okay. Yes, I just wanted to emphasize on the courses that you spoke about, yes, that is available on the platform for additional or reinforced learning. So for some of you that already have a background in molecular biology, I mean, that's a plus for you. But I would still encourage you to go through those courses because one, you would be required in between or at some point in the training to use the T-Bio platform to execute some assignments using data that were made available. And you will find some of those courses of immense benefits when executing these assignments. So I encourage you to really take them seriously at your pace, obviously, and enjoy while you're doing that. And then that also helps when we come back for another another meeting for questions to be asked, and we can help you with those questions. Okay, so and just to reinforce again on the type of computing you need or data space, everything you're working on in terms of processing will be in the cloud. So you don't need any extra space on your computer, all right? And you would see that once you start, especially on the T-Bio platform. Some of the processing of the pipelines, which is just the steps involved in you processing a particular set of data. Some of them could take a short time, some could take a little long, probably like up to 30 minutes, but essentially all this data is up in the cloud. And so you don't need any form of extra data space on your system. Just wanted to reinforce that. Thank you.
Thank you very much, Ma. So Mr. Oluwatobi, you're also raising up your hand. Mr. Oluwatobi, are you still there?
Yes, I'm here. Good evening, sir. Good evening, everyone.
Good evening.
So thank you for this session because I was aware of this program and I was a little bit skeptical. So my question really is, this program, would it be enough for you to, I mean, for example, I'm in intern at NIMA and we run some sequencing sometimes and we generate data from our sequencing platform. And there's a particular one we do, HIV resistant. So we use all these kind of applications that, you know, just give it to the application, click, and before you know, it brings something out. And you don't really know how it's doing that and all of that. Like that one can only do for HIV. You can't do for other organisms or some other form of analysis. Are there any limitations to this particular training, like in the scope? Is it that it can only work for a particular kind of research, or you can apply it to like a general kind of research, antibiotic resistance, searching for mutation, searching for variants, and all of that? I don't know if you get what I'm saying.
Yes, Mr. Oluwatobi, I understand it perfectly, right? You know, we said this is an overview into bioinformatics, right? And our platform has all these bioinformatic tools. For example, as session 2 and session 3 will be on genomics mapping, visualization, and variant calling, right? We actually, if you are familiar with bioinformatic tools, you know that when you want to call, when you are doing variant calling, you're talking about SAM tools, BAM tools, you're talking about Bowtie tool, you're talking about BWA, you're talking about what am I talking about? All these other bioinformatic tools. Every of them, we have them on our platforms. Whether you want to use it for metagenomics or infectious diseases, whether you want to use it to understand cancers. In fact, by our second session, we actually visualize COVID data, COVID sequencing, we visualize Ebola sequences, right? We know that for now, it's very cheaper to sequence viral or infectious diseases. The last time I tried to get a sequence for human genome, and you can check, it is still $1,600, right? You can check at Redeemers a day. Some people you have to probably import it. Now, imagine a researcher, and you're already paying $1,600 for just one person. So if you want to sequence 50 people, you know, that's already all the money for that. And you know, especially in Africa, in Nigeria, where most research is self-funded, that's a lot of money, right? So what our platform has is, we also teach you how to get those sequences. You don't need to sequence them yourself, right? But even if you sequence them yourself, on our platform, you can visualize your sequences. You can map your sequences to any reference genome of your choosing. You can understand, you can do variant calling, you can understand mutations, right? You can, aside that, you can go further and understand SNPs, and understand causes, either genomic or transcriptomic level. You can also go ahead and do what we call differential gene expression, while you understand the differences in gene expression of this. So all this, it is not related to one discipline, right? We've had colleagues that are microbiologists, medical lab scientists. As I said earlier, I'm a clinician, I'm a dentist. We have medical doctors, we have any aspect of molecular biology. The doctor is a molecular biologist, and for every aspect of life science. Not only that, even for machine learning, right? On our platform, I'm sure you've got a lot of machine learning algorithms, like random forest, like decision tree, like anything you think about, everything is available on our platform. And you can use it for any of your research, right? And as I said earlier, because—
One more thing.
Yes, sir.
Okay, so concerning the computer requirement, I think I have the computer requirement, and I want to be able to do it on my laptop, so that in such a way I'm not really dependent, in case I'm done with the course and all of that, and I need to do it on my laptop. I'm not really dependent on a server, kind of, because I have some software too, and I don't know if there's provision for that, so that I can do it on your own laptop, your device. I think I have the computing power.
No, if you have, that's what I'm saying. If you have the computing power, right? We have a platform that will make it easy. But if you have computing power, then most of these are won by programs, right? We'll teach you, and that's why we said there's nothing you will do on genomics that you can't do in R or Python, right? We'll also teach you R and Python in case you want to do it yourself on your own system, right? Not only are you going to, you can do it with our, what's it called, our T-Bio server, but if you also want to do it, if you want to understand the basics of this, the fundamentals of it, and do it by yourself on your system, we also teach you the basics, right? That I said, R and Python learning is part of this program. We introduce you to it, right? Even some part of Linux, we teach you, Jonathan, as part of this program. But don't mainly,
Linux.
What do you say?
I have Linux and Ubuntu.
That's what I'm saying. We'll teach you how, we'll teach you Python. And in going through this program, once you understand the basics and why you do these things, you can do it by yourself, right? The only thing we are saying is you have, instead of you to require that computational that computational space, our resource is cloud-based. You can just put it on the cloud and go and do your thing. But every fundamental of each bioinformatic algorithm and each bioinformatic tool will be taught from the basics in this course. Do you understand me, Mr.?
Thank you so much. Thank you.
I also use a lot of Linux. I use a lot of Linux. I use a lot of R. I use a lot of Python. But I also understand that for each bioinformatic tool, you have to learn, probably dedicate 2 to 3 months to learn each tool before you can begin to work, right? We are circumventing that you can learn, you can continue doing your project as you learn. You don't need to wait for 3 months to understand R, or 5 months to understand Python, or 6 months to understand Linux before you begin to work on a project, right? We can learn as you practice. That's our goal. Thank you very much. So I've been saying a lot about this T-Bio server. Let me just go straight and show us how it works, or show us its basics. So I'll do that in a minute. Let me. Okay, can you see my screen?
Yes, we can.
Okay, so this is our second platform. We call it the server tbio.info. I'll put the link also here, so anybody can also go straight to it. Right. I want to show you the whole extent of this, right? So this is the Bio server, right? And you can see that it has a lot of options, a lot of functions. As long as you are dealing with next generation sequencing data, there is no function you can't do on this, right? This is Transcriptomics. This is Genomics and epigenetics. This is just working strictly on DNA and RNA sequences. If you want to work on microRNA, this is Metagenomics and microbiome. This is CHIME2 for people that know Metagenomics, right? This is also, you can also treat your mass spectrometric data here if you are dealing with proteomics or metabolomics. And if you come to this side, you can also see that there are data mining and data learning features, right? And all of these can be done at just the touch of a button, right? All you need to do is just click on this and to take you to the next stage. One good thing about it, one lovely thing about it is AI enabled. So once you click on a pipeline, it immediately gives you options, right? Because I said there are a plethora of ways to analyze data. But once you click on the pipeline, it automatically tells you the next 1 or 2 steps to take, right? And guides you until you reach the end of that pipeline. For example, we also have demo data sets that you can use to learn, right? You can use this to learn, especially if you are not a registered participant, and yet you want to have a feel of this platform. You can use this data set, right? These are RNA-seq pipelines. The breast cancer pipeline I was talking about, this is the PDX, right? If you click on it, right, you can see bringing you to these sessions, right? And if you check on here, all of these are what are bioinformatic algorithms. I won't like to go into them because, you know, as I said, this is like a beginner program. Some people might not have an idea of what this does, right? But if you are familiar a bit with bioinformatics, you will know that all these tools by themselves are the common bioinformatic tools. You have Clean Primer, PCR Clean, Genomics, and instead of you running commands on your Linux, on your Python, to be able to get the solutions to this, all you need to do is just click. For example, if I click on start, like I want to start.
Don't waste time, I beg, please.
I want to start a pipeline, right? Here, I can see my input files. You know, I said earlier, these are demo pipelines. They've been imputed onto this, right? So I just click OK. Then, almost automatically, you can see that some tools are highlighted in yellow. And I said, this is AI enabled, AI guided. It's giving me options. Now, out of all the plethora of bioinformatics tools, the next one I need to use is any of this, right? That's the beauty of it. So if I just click on this tool, just at this button, what the first thing I see, it will explain what that tool does, right? The Genomics is a tool that is used to trim adapter sequences, used to trim poor quality weights on a sequence. So it's showing me what it does, right? And if I click OK, it will do that action without me pressing any commands, right? You can see that after clicking this, other things are also being highlighted. Here, it's only the PCR clean that's been highlighted. For people that are familiar, all these steps will take during the course of this program. But I just click on this PCR clean. I'm saying clean the PCR duplicates that are present in this sequence, right? I'll click OK. You can see that it's showing me other tools, right? And that's how it will be able to guide you from one bioinformatics tool to another. From here, it's highlighting just these three things, right? I can then click on one or either of them to the end of the pipeline, right? That's the beauty thing about this. And once I click on it, for example, let me click on this. So let me just click OK. You see, for everything I click, it tells me what it does, right? I go to the next one. Once I click on this, it tells me what it does. Then I can end the pipeline, right? And when I end the pipeline, what do? I continue my day. I don't need to wait for it to finish. Some are immediate. Some will take time depending on what you're working on, right? But that is it. And I can get my results without needing to have computational size on my system or have the expertise on how each of these things work. I don't know how PCR clean works. I just click on it and it does its job. I don't need to know how Genomics works. I need to know what it does. We'll teach you what it does. But I don't need to be an expert in Genomics, right? And it does its job and I get my results. That way I'm doing projects, I'm learning, and I'm doing projects, I'm getting answers, I'm analyzing things, right? That's the beautiful thing about this server.tbio.info. So starting from next Wednesday, we'll teach you about the in total of each. What do you do with genomics? What functions do you click? What does pre-processing mean? What does mapping on genome mean? What does mapping in transcript mean? What do you mean by differential gene expression, right? All these tools and their functions at the beginner level will expose you to it, right? I also like to pause again and ask, are there questions before we go on to this program per se and just run through? Questions?
Please, sir, if I may ask, is it instructor-led or it's self-taught, like if we are on the...
Instructor-led, right? And like I said, it's more like an hybrid session, right? We'll have those educational resources where you learn the basics, fundamentals, self-paced, right? But on Wednesdays, we'll come together, right? Where the mentors, Dr. Toye and Hai, and other international mentors, will be here to guide you through those sessions, right? We'll do everything together here. Everything we're talking about, we'll do it together in this session. We'll give you the video and we'll be available all week, either on WhatsApp, on Slack, or through personal calls, right? You ask us and we'll guide you through. That's one of the advantages of it, that it's mentor guided. When I started learning bioinformatics, I didn't have a mentor. I was just learning everything, do you understand? But when I joined Pine Biotech, there are mentors, there are local mentors, there are international mentors, and they are all available at any point in time on both our WhatsApp and Slack channel. Just come and ask any question. Somebody will answer you. And that's the good part of it is every point you have someone to ask a question about a clarification. Did I answer your question, sir? Hello, sir. Please, are you still with me? I hope I'm not alone here. Well, thank you, Mark.
Here with you.
Oh, okay.
Yes, you did. Okay. Thanks a lot, sir.
Okay. So it's mentor guided. It's absolutely mentor guided, and all our mentors are available at any point in time. You can just give us a call, chat us up on. You can throw your questions in the WhatsApp, throw your questions on. We're going to have a Slack channel where you can also ask questions, and we'll answer your questions. both local mentors and international mentors, right? And as the other earlier speaker said, what if I want to do it myself? What if I don't want to use our platform? And I'm like, okay, we also teach you the programs needed. We teach you how, from step by step. We teach you Python. These are the programs that you need to do it by yourself, right? Those are also available on our Learn platform. And another thing we also do is, for every session, we just won't go home. We also give you assignments. I will tell you what to do. For example, there's already an assignment, even though this is a free, we already have assignments. You can see you already have 4 assignments, right? You have to complete your profile, like I explained earlier. You have to explore the server.info. If you're interested, just send us a message at omixlogic.ngl@gmail.com. I'm typing it. We'll send you a demo username. Yes, gifts, homework, yes. Adult education, you'll be tested. As I said earlier, it's practical hands-on. This is not just theory. There is no theory in bioinformatics. It's practical hands-on, right? So the assignment for today is already 4. You have to complete your profile on the platform we first talked about. You have to explore server.info. We send us messages, myself and Alfred. We'll send you a demo account, right? That you just enter, just click around, just walk around, see what it does. Free of charge. You can use any of the demo pipelines to test and see. Then we already have 2 courses that are absolutely free. Introduction to Bioinformatics course, which I'll try and write the link again. I think I put it there already. And also Introduction to Bytes and Molecules. These are 2 courses that are absolutely free, and they are beginner courses, right? By the time, for people that will register, this is the first thing we'll ask you next week Wednesday, that, oh boy, oh lady, I hope you have done this, right? So now to the program itself, the Introduction to Big Data Bioinformatics, which I said earlier, in 6 interactive sessions, we're going to talk about what is next generation sequencing, what is biostatistics, what is big data analysis, right? We'll be able to learn about these multi-omics and learn how to apply them in solving questions in your field, right? The first session starts next week, Wednesday, November 24, at exactly 4:30, where we introduce you to big data in biology, right? What started this data explosion? Next generation sequencing. What is the evolution of sequencing? What are the generations? The first generation, second generation, and third generation. What are the steps needed for next generation sequencing? Right. Then the week after, we'll talk about introduction to genomics. We'll talk about DNA variant, mutations. What does it mean? How do we do variant calling? What is somatic mutation? What is the what does somatic SNPs mean? Single nucleotide polymorphisms. How do they characterize cancers or molecular pathogens of cancers? We talk about codons, amino acids, what are genes, right? Then we're able to visualize our sequence, right? When you sequence a data, how do you even visualize it, right? This is a picture of a visualization website known as JBrowse, right? And how do you understand? Then later I'll go to how do you analyze this genomic data, right? As I said earlier, when I showed you the platform earlier, I said we'll teach you what each of these steps means. What do we do in processing? What do we mean by mapping alignment? What do we mean by variant calling? What do we mean by annotation? What do we mean by differential variant calling? How do we understand and analyze this? How do they help in learning about or analyzing susceptibility to infections and disease? So that will be our second topic, right? Then our third session will be about transcriptomics or gene expression data analysis. I'm sure for molecular biologists, we know about the movement from DNA to RNA to proteins, right? The total mRNA in a cell is known as transcriptome because they are called mRNA transcripts. We understand how gene expression data analysis can help in understanding diseases and infections. Max, no problem. We'll send it. Just drop your email address, or as I said earlier, just send me a mail at omicslogic.nigeria@gmail.com. The recording will be available for all, right? We also post it at our Pinebao YouTube channel. You can also watch it there. And that, you can send us a message at omicslogic.nigeria@gmail.com. So this will be our third session on transcriptomics, right? We'll teach you how do you prepare data for analysis, right? We'll also teach you how to make use of this platform to understand this. As I said earlier, our AI guided platform is color coded to be able to guide you to talking about the next thing. So this is what we'll do in our third sessions. So as I said, we also show you examples of the breast cancer cell lines, right? Why do we use cell lines? When do we use tissues to understand whatever we need, right? The next session after that will be about differential gene expression, where we use statistical approaches to study and understand RNA-seq after mapping them to reference genome. Then our fifth session will be on statistical analysis and machine learning. We use statistical approaches to study the data we had gotten from our gene expression, right? We'll start finding relationships in data, and we'll collect data on different variables and compare them with their gene expression data, right? And we'll teach how to get that done. Then our last, well, I said part of that, we'll also teach you data analysis in Python and how you can visualize your sequence. And our last session will be introduction to bioproject, right? Because of the high cost of sequencing, how can I get these sequences? How can I get, because most of these sequences are being deposited at public repositories, where they are publicly available. How do I search for sequences? If I'm working on A, how do I get sequencing data for A? How will I glean it? And how do I start to analyze it to be able to gain information for my own research? That will be what we'll end up with. As I said earlier, on our platform, every achievement gets you points. If you complete a course, 500 points. If you run a pipeline, 100 points. For your profile, and all these points are added together to get you points, and which will get you certificates, right? So this is just a rundown of our program, the Introduction to Big Data Bioinformatics, that starts next week, Wednesday. At this point, I would love to pause and entertain more questions. Hello, everyone.
Hello.
Yes, I said, are there questions? Before we go straight to the last thing we'd love to do today, and is registering you for the program that starts next week. Any questions?
So to register for the program,
yes.
Do I need to make the payment first before the registration?
The payment is actually the registration for the program. Before, we had one report to registration to allow you to be part of this program. But to be part of the program itself, the registration is by payment for that program.
So, and you said it cost how much, please?
I didn't hear that, ma.
How much you said it cost for the program?
Program itself is $45, ma.
All right.
Okay. So I want to ask, is there a way we can pay one of, like, from start to finish, you pay instead of paying in bits?
No, we don't. No, that's what I'm saying. I say for this program, it's an independent program, right? This 6 program is an independent program, but is more like a beginner program, right? We plan to hold series of programs later. Some are even as lasting as 3 months program, right? So we feel that it's more sensible, it's more economical to pay for a series of those programs, right? And it depends on the number of participants that will decide on how often do they want the program. Is it quarterly? Is it? So for now, we are just holding independent programs. Just understand. So for now, this program is a standalone, right? Even though it's by the next program, it will be another level for this. But this program is a standalone program for now. Dr. Toya and I are working on an exhaustive, comprehensive semester-like program. When that is done, you can then say, okay, pay for 3 months. We'll teach you from A to Z. But for now, this is just an independent beginner program.
Okay, I would like that. I will register for this, but also I would also like that kind of 3-month thing. So because I would just want to pay one off, and I'm focused on instead of I think I'll still prefer both.
Yes, that I'm saying, we will keep you updated since we have your contact details. We keep you updated about all our programs, the beginner ones, the advanced one, the intermediate one. And when we have a semester-like course which we are working on, we also let you know about it. Any other questions? For this course, yes, payment is the payment is registration gift. The payment itself is the registration. So when you register, you'll be let's quickly go to the program page. So you understand what I mean by that. So I'm putting the link here so that you can see the link page. Sorry, a minute. That's a mistake. So this is the program website, right? When you come here, this is where you get information about the programs, right? You get everything. These are the mentors and faculty. This gives you this is more like the program syllabus, right? And when you click on register, if it was before this program, we had this to more like a pre-registration page for this free webinar, right? But now at this point, if you want to register for the program itself, just give me a second, please. I also put this on the chat box. So the second one takes you straight to the payment page. So I think my network is a bit slow. I apologize. Okay, so this is payment. This is the program page too. So when you click on start your application, it brings you these options, right? $45 for this for most people, and it has all this, right? If you want, aside this, if you want one mentor support, you want us to be putting your hand and guiding you all through, you can pay for $60. Or this is if you want to, that even after this program, you want us to assist you with a project, right? You can pay this. But for most participants, especially for beginners, you can pay the $45 where you click this select option, depending on what your preference is. So once you click on this, it will open our payment page where you can input your details. So I hope you can all see my screen.
Yeah, we can see it.
Okay. So you just put your email address, which you already opened with us, put your card details, and click on pay, and you'll be registered for the program. So once again, let me put this on the chat forum. This is the full payment page. Once you click on this, it brings you directly to our payment page where you just click on start your application, enter your card details, right? Start your application, enter your card details, right? After selecting any option you want, and you'll be registered for the program. The program starts fully next week, Wednesday. where we're going to have our first session.
Sorry, sir. I think it would be nice if there's a Slack channel immediately where we can have this.
Yes, for registered participants. For registered participants, we have a Slack channel where for all participants, we actually have a WhatsApp channel where you can get more information about the program. But for registered participants, we also have a Slack channel where we add them to. That way, we can you get questions. All our mentors are added on that platform. So as you're asking questions, local mentors, national mentors, they can assist you with your questions, right? But we also have a WhatsApp channel, which is just for all participants. This, like for example, this video we posted on that channel, right? Also, we also email it. So we make use of all available channels to get across information. We have WhatsApp, we have Slack, we also send emails. So, but the informations, and we also ask you to, I'll post it on our WhatsApp channels later. We also encourage your indulgence to follow us on LinkedIn, Instagram, and Facebook. We have all this information where we put it. In fact, I remember for the people that did pre-registration, I sent them WhatsApp links to join our WhatsApp. But for the registered members, we send them a Slack channel where they can join, but strictly for registered participants. Any more questions? Any more questions? Is Dr. Toye still around? Dr. Toye? So I'm also putting our WhatsApp link on this chat forum. So you can just click and join our WhatsApp group and you get more information from it. As I said earlier, you can contact us at omicslogic.ngl@gmail.com. We'll answer all your questions. If there are more questions I would love to answer. If they're not, we would love to thank you for being part of us today. No, you don't have to pay today. You have till next week, Wednesday, to pay. Yes, you can pay before next session. Please, you can pay before next session. We'll gladly, and even after next session, you can pay even before the second session. We'll send you the session video of what you have missed, and we can start from there. So you still have opportunity to pay before the next session or before second session. We stop accepting after second session anyway, because as I said, this is a computationally comprehensive program. We don't want to strag people that won't be able to work with us along. So you have a chance to pay to the second session, right? If there are no more questions, it's been an eventful evening. I'm really elated. I thank everyone that's come for this program, for the time spent with us, for your indulgence, for your questions. I hope you will be part of this program and we can create life scientists. Greetings from myself, from Alfred Akinlalu, and from Biotech in Nigeria and globally. We'll make sure to send the videos over to you for this session, and we expect to be part of this program. Thank you very much, and we'll see you soon.